Good morning to all. I am Dr. Sundeep Kumar from ICR MEPR New Delhi. I shall give my presentation on identification and validation of novel QTLs for seedling and adult plant resistance to stem rust. We all know the importance of wheat as a staple food. This is the largest primary commodity. More than 2.5 billion pupils in 89 countries depends on wheat as a staple food. There are a number of challenges for the successful wheat production. Stem rust is one of the major challenge for the successful wheat production. Though more than 60 SR gene, resistant genes are known, number of them are being used in breeding programs. However, this UG99 and its variants has made most of the resistant genes susceptible. This Deglu and Sicily race, which were earlier there, they have again reappeared in Europe and UK. And the predominance of this PGT race of stem rust is responsible for large regional epidemics and crop losses. So keeping this thing in view, there is a need to, evaluate the germplasm lines for the novel seedling and field resistance against a contemporary spectrum of PGT races. Due to the global spread of the elite wheat germplasm, our indigenous wheat germplasm lines has been neglected for a long time. It has not been evaluated for the biotic as well as abiotic stresses, and it has not been utilized for the identification of the novel genes. Keeping this thing in view, what we did, we used a panel of 400 diverse germplasm lines for the identification of the QTLs for seedling as well as adult plant resistance to stem rust. We did phenotyping for seedling resistance as well as adult plant resistance. We did genotyping using 35K exome array. We use this 35K data for the to know about the population structure. A diverse panel was there, and this diverse panel using different models. We identified the QTLs and the genes, and we validated them through the CAS markers. We evaluated this diverse panel of 400 lines under the field conditions at IRI Regional Station, Wellington, and for seedling resistance against 4 different races at ICR, IWBR, Flowerdale, Shimla. We got, this moderate to low level of correlation among different environments of pathotypes. Out of this 35,000 SNPs, only 18,932 SNPs showed polymorphism, and they were distributed on 21 wheat chromosomes, as listed over here. These 400 lines were really diverse in nature. We got 4 subpopulation in it with some admixtures, and it was further confirmed with the help of NG tree analysis. And this PCI analysis also showed that these lines were from the different geographical regions. This circular Manhattan plot shows the significance of the association of synapse using multi-locus mixed linear model in response to system rust at seedling and adult plant resistance stage. We identified 20 reliable QTLs for seedling and adult plant resistance. This is a summary of the markers which we identified for seedling resistance against 4 different races, as mentioned over here, and they were contributing, significantly towards the total phenotypic variance. Similarly, we identified the QTLs for adult plant resistance following all 3 models, and they were consistent across 2 or more environments. The significant markers were further validated with the help of CAS markers. We got, this promising stem resistant lines for adult plant resistance as well as for seedling resistance. There were some lines which were highlighted by the red colors. They were having more than 18 favorable alleles for stem resistance, while the lines highlighted by the pink colors are those which were showing the resistance not only against adult plant but also against seedling resistance. So in conclusion, we can say the identified QTLs are contributing significantly to the total phenotypic variance, and the 3 QTLs which we identified on 6AS, 2AL, and 7BS have been validated using CAS markers, and one of them is which was on 7BS was effective at seedling as well as adult plant stage, And these QTLs have the potential to be deployed in wheat improvement programs to develop disease-resistant varieties for seedling, as well as adult plant resistance to stem rust, and can diversify the genetic basis of resistance. The germplasm lines which we have identified resistant can be used as a source for developing stem rust resistant cultivars. Thank you.